I see attendees are joining.
Should I share my screen and put that on already, or?
Yeah, I think that would be good.
Oh.
Can you share your screen or not?
Yeah, I can.
Okay, good. I see attendees are joining. Maybe let's wait 1 minute and then start.
Okay.
Okay. Hello, everyone. Thanks for joining this webinar today. We have Dr. Kohitij Kar as our invited speaker. Dr. Kar is an assistant professor in the Department of Biology at York University. Before that, he was a research scientist at McGovern Institute for Brain Research at MIT. And he received his PhD from brain and behavioral sciences at Rutgers University in 2015. And Dr. Carr's research is interdisciplinary at the intersection of vision neuroscience and artificial intelligence. We are very excited to have him here today, and he will talk about his exciting research at on the role of recurrent neural circuits in the visual cortex. Dr. Ko, you can take the stage and start.
All right. Thanks, Yalda. Thanks for the introduction and inviting me for this talk. I can't see anybody, so this is going to be a little bit awkward, but I'm hoping that you will enjoy the talk and we'll have a lot of questions. And so feel free to ask the questions in the Q&A.
Sorry to interrupt you. Everyone, you can use the Q&A function to ask your questions. You can type your questions throughout the talk, and we will discuss them after the talk. Sorry that I interrupt you.
Yeah, no problem. That's good. Yeah, so feel free to ask your questions, and I'll be happy to take your questions later on. So, okay, so today I'm going to talk about this is mostly my postdoctoral work. So I'm going to acknowledge Jim DiCarlo at the very beginning because most of the work that I'm going to show was done in his lab. So as you see, the title of my talk is Probing the Role of Recurrent Neural Circuits During Primate Core Object Recognition. So why am I using all these words? And these words have no meaning unless I actually operationalize them, really. So because they can mean different things to different people. So let's jump directly into the talk. So first, I want to kind of tell you a little bit about the research program overview that I have in my own lab and why we do we do neuroscience in a very specific way. And many other labs follow this same formula, but I just want to go over it a little bit in terms of motivating how we actually ask questions in the lab. So why study the brain? And why don't I study toenails, for example? I am particularly interested in studying the brain because the brain supports a lot of our remarkable behaviors that we are very dependent on, and our life will really suck if those behaviors start disappearing. So that's why I think I at least study the brain. So when we want to study the brain because of behavior, and we want to really quantify these visual-cognitive behaviors. So we in our lab study a very specific type of behavior, which is mostly visually driven, which is what I would talk mostly about. So I'm going to give you an example of one type of visual behavior, which is core object recognition. And it's a part of a very rich regime of visual behavior that we demonstrate. So here's an example. So imagine I'm walking through Fifth Avenue, New York, and I'm making small eye movements to fixate at particular locations and tell what information is there. So if I fixate here, I can tell there are two people. They might be too close to each other. You might infer that they're friends. Here's another person who might be looking a little bit confused. Somebody has raised their hand, but I guess we'll have questions at the end, right, Yalda? Okay, cool.
Yes.
Here's another person who's, unless there is some technical difficulty, so maybe we can, you can see that, but like other than that, we'll keep the questions until then. Here's another person who is looking at his phone. We move our eye again. He's another person who's looking at their phone. We can do things like we can quickly tell time. We can look at, food, and we can, okay, this might trigger some memory, previous memory of what happened when you last ate that food. Anyways, so we can do this very quickly, and within like hundreds of milliseconds, we can tell these things apart. And this is what a lot of folks in the field has defined as the core of object recognition. Happens very fast, foveally determined. So we study this very critical behavior that we do seamlessly. You did not think much about actually discriminating these objects, but it has been through decades a very difficult problem to solve. So then we ask, like, how does the brain support this behavior? This is part of the whole closed loop. And to ask how the brain supports this behavior, we have to do two things. First, we have to probe brain mechanisms. So usually we will use some technique to actually directly interact with the brain and probe the brain and see what's going on inside the brain. But also the how question is answered by increasingly building brain-like computational models. So that is that these two actually together solve the problem. Just probing the brain is not going to solve the problem. You have to also give an answer to how the brain works. And oftentimes these answers are in the form of this computational model. I just want to take a brief moment and ask the question, like, why is this important? Why do we need to ask this how question? Many people have many different answers. Some might be driven by, the sense of discovery of like, I need to know what is happening. I will argue that most of it, implicitly or explicitly, is taken, people undergo these efforts for clinical translation. It's for either you are in denial of it or you explicitly make it your claim. So we in our lab, we explicitly make it our claim that we're going to try and impact clinical translation. Okay. All right. So there are, when you want to increase, there's another how question is that how are you going to improve these models? What are you going to do? So let me just go through what knobs do we have for actually probing the brain. Okay. So in terms of knobs, we have two knobs in our lab. One is the behavior. We can ask human subjects to do different behaviors. We have a monkey lab, so we also ask monkeys to do the same behaviors. We have our own home cage system where the monkeys can do this. The interesting thing about, or useful thing about, the monkey as an animal model for human vision is that you can also probe internally the brain of the monkey with electrodes. You can record their activity, you can perturb their activity, so on and so forth. So at the end of the day, anything that we call progress or knowledge or mechanism understanding, the answer to the how question, it boils down to what kind of models of the system are you producing. So what is your current best models? And these models have evolved through time. So for instance, there were models called HMAX, computer vision model called SIFT. There are obviously word models. Many people would say, you'll hear brain area X does face processing. This is also a kind of implicit model. That model runs in the head of the scientist that is telling you that answer. So anyways, there are different kinds of model. And by the end of 2014, the type of model that was most prevalent that really came to the stage were this feed-forward deep convolutional neural network model. So they became kind of these models that could explain a lot of this behavior and neural phenomena. And I'll go into this a little bit further. But I first want to, maybe I'm just reiterating the things that you have heard during the course or in general, but it doesn't hurt to reiterate this point that why all the hype. The main reason for the hype is that for the first time around 2012, 2013, there were models that were very accurate in actually solving this problem that, on the surface looks very simple to us, which is that you see a dog like this and you transform this information through your brain and you call it a dog. So models were supposed to do this, but they struggle because you don't really call this a dog. You also call this figure a dog, this image a dog, this image a dog, this one a dog. You have to call all of them. You have to classify them at the same level as dog, which was not a very easy thing to do. And over the course of time, people have solved it now to a certain level of accuracy. The another quite striking thing was that this process of solving this in the deep convolutional neural network field has been deeply influenced by the architecture of what we know, the architecture of the brain. So for instance, the primate brain has this very nice hierarchical structure in its ventral visual pathway where the information flows from V1 to V2 to V4 to IT. And this pathway is kind of mimicked. People have found, researchers have reported that this architecture is kind of mimicked in the best neural network models that came up initially. Things have diverted quite a bit now, but this was at least the initial discoveries that were made. So 2014, V4 and IT were associated with the model, were predicted by the models, and recently also early areas like V1 and V2 have been mapped onto this model. But these models are not complete models of the system. They have problems. So this is a plot from a paper that I'm showing you where you can think of the higher bar as like humans predicting humans or monkeys predicting monkeys in their own behavior. And the bars are how well the models can predict this behavior. So what I'm trying to show you here and motivate is that there is an explanatory gap. These models cannot completely predict the behavior of the human. So we need to improve them, right? Okay, so how to bridge this gap? Again, this is another plot. Don't worry about the details. The main idea is that the neurons predicting neurons. So here we are building models that are also models of the internal components of the brain, which are the neurons. And the state of the field is that there is still an explanatory gap, means that models cannot predict the neurons as well as the same neuron can predict itself for a different trial of an experiment. So these explanatory gaps exist, which means that we have to improve this model. So how do we improve this model? What is missing in these models that we can improve on? So in the rest of the talk, I'm going to give you an example of like how we went about this problem and how we tried to find a solution to this problem. So let's first, what we first did is let's try to like come up with some things that we know about the brain that we clearly also know is missing in the model. So for example, the brain is dynamic. Most of these models are static in nature. They don't have any temporal dynamics. There are other visual areas than V1, V2, V4, IT. There are recurrent connections in the brain. Brain has a nice functional topography that has been reported for a long time. Models don't have them or did not have them. There are models that are beginning to have them. Learning mechanisms in the brain are known to be very different from the learning algorithms used in the model. Brains use dendritic computations. There is no notion of such structures in the model. Neural noise correlations, a lot of stuff. By the way, one sort of nuance point here is that a lot of these computationals, although they are not explicitly put in the model, somehow the model might still be capturing those. And those are interesting research direction to pursue, at least in my opinion as well. But anyways, for me, I selected my favorite 3 items. And I said, okay, let's start with these items and let's actually start to ask questions about recurrent connections. Like, can you put recurrent connections and make the model a better model of this core object recognition phenomena that I'm explaining to you? Okay, so how do we, formulate this as a study? So as a study, we have 2 systems. We have the brain of the primate, we have the primates as a system, and we have the models as a system. And I start from the premise that these are not equal. They're not equal, they're not producing the same behavior. So the first is the motivation. So, okay, we start with the idea that primates have a feedforward system, and so do the model, which is what I already told you. The missing part is this recurrent computation that primates clearly do and models don't currently. So our question was that, are these recurrent circuits functionally relevant during this sort of core object recognition behavior? Are they good? So of course there are these recurrent connections in the brain and they will be useful for something. But to make the problem tangible, you have to actually quantify the behavior and make sure that your problem is actually relevant to the behavior that you have actually quantified. Because otherwise it's sort of, you can do whatever and that will still make sense, which is not the best way to do science. So the first step is compare behavior. Can we compare the behavior and see whether these 2 systems actually differ in a very specific way? What is the behavior? So we do this very simple behavior where we show an image and then we show 2 options. And we say, what was the image of? Was it a face or a car? Here's another example. Was it a bird or an elephant? And just with this very simple behavioral paradigm, you can come up with many, many types of trials if you use different objects, different images. So we use thousands of images. We use synthetic images where we have full control of where we are placing the objects. And we also use natural photographs from data sets, image sets like Microsoft Coco. Okay, so here's an example of what a comparison of behavior would look like. So each dot is an image. And you can see that there are many images that line up in the unity line, which means that if a model is good, humans are good, or a pooled monkey performance here, what I'm showing on the y-axis is good. If the model is bad, the monkey performance is bad. So they're aligned with the models and monkeys are similar. This is what we call control images. And there are many challenge images where the monkeys are much better than the model. And our hypothesis was that maybe some of these images, the monkeys are better because they have some machinery that the models do not have. And one of those pieces of machinery could be recurrent processing. So that's the hypothesis. And to see whether to basically test whether we have some evidence to support this, we have to look inside the brain. So that's what I did next. We implanted arrays in known areas of the brain, that are known to have the solution for these objects. And so we implanted arrays in area IT, and we asked, are there any systematic differences between the processing of challenge images versus control images? At least that's how I'm going to, explain this study to you right now. So these are the Utah arrays that we implanted in these different areas, and then we were recording. So I'm going to give you an example of what this whole recording and analysis basically entailed. So I'm going to talk about a very specific behavioral band. So in this small behavioral band, you can assume that challenge images and control images have the same behavior as far as the monkeys are concerned. So let's pick one image from the control image set, and let's see what we do. So this image we show to the monkey, and we are recording from the monkey's brain, and there's a lot of activity in the brain. So what we first try to do is we try to construct this thing called a population vector, which is just simply, you can think of this as counting the number of spikes per neuron, okay? And you can build a decoder from these population vectors that try to decode out what kind of object is there in the image that is shown. And this sort of gray brownish band, this is the monkey's own accuracy. So it's pretty high for this image. So here I'm going to show you, it's going to make sense soon. When I go through time, so right now it is at 0 because the image has just been shown. 0 means the image has just been shown. The information hasn't even reached IT yet. If you go across time, at some point, the neuron starts to fire. These are the green, signs here. And then the decoding accuracy starts to rise, and at some point it hits the accuracy of the monkey. So this is almost like, I'm reading the brain of the monkey, and I'm seeing how quickly is the information evolving. Or it's not a comment about how the brain is doing it. It's a comment about what I can read from the brain. So these are interesting sort of differences that people often confuse together. So this has nothing to do with how the brain is solving it. It is how I am able to solve the question that I'm posing using the data of the brain. But it still gives us some interesting ideas about how the brain might be solving this kind of stuff. So here I'm showing you that this particular image almost hits solution around this particular time. At this time, the solution that I'm deriving and the solution that the monkey is reporting way after this time period is actually the same. So here's another example I can pick from Zebra, shows the similar timing. What was interesting to us was that if you pick the challenge image, the timing was slightly delayed. So there are two things here to observe. One is that the decode actually got delayed and started to rise much later. However, the neural responses were still quite active around the same time, which I will show you a little bit more later. Here's another example of a dog that also gets, it starts developing slightly later, but again, the neuron starts to fire at the same time. So if we look at the neural responses averaged across all neurons over time, for both control and challenge, there's no difference in latency. It's like some kind of secret sauce comes into IT later on and allows IT to linearly decode these representations into object spaces that are easily categorizable, right? So this sort of starts to develop, at least for us, it started to motivate the idea that maybe this is what recurrent processing is doing. It is making whatever, wherever the feedback is coming from, it is trying to make the solution more linearly separable at the level of IT. So that was the idea. There might be other explanations to this. We did a lot of control, analysis to rule out multiple different explanations. You should read the paper for that. But we did, measure this object solution time across multiple images. And I'm just showing you here 16 examples. And overall, there were around 30 millisecond delay in the distribution of these two sets of images, control and challenge images. And so we think that this additional time, compute time might be symbolic or might symptomatic of recurrent processing that is happening in the system, and that the system is actually leveraging to improve their performance on those challenge images. There was another sort of interesting observation that oftentimes in this field, and maybe you have gone through this in some of your coursework, that models, these feedforward models are also used to predict the neural responses of the neurons that we are recording. So what we observed that if you try to predict the early responses, those were better predicted than the late responses. And oftentimes this late activity that I was showing you was, associated with recurrent processing. And so it is sort of converging evidence that maybe these models are specifically failing to account for the part of the response where there is more feedback information in the system, which the models are currently not accounting for. Again, there was a lot more analysis done on this at our end in the paper, so please read the paper if you're interested. So this explanatory gap is actually also could be considered another sort of evidence towards the fact that the models are missing recurrent processing, and that recurrent processing is actually really helping the system to gain higher accuracy. Okay, so what are we doing apart from this? So once we know that, so the main inference from this study is like, hey, deep neural networks maybe need recurrent computations to solve this problem in a brain-like way. Of course, the computer vision community does not need to care about this inference, because maybe they come up with a different kind of solution that they might make they might invent transformers, or they invent deeper networks. So that's not their job to make a brain-like model. It's the job of the neuroscientists to care about the brain and make brain-like models. So for us, we thought, okay, can we make these models? And this is a collective effort where I was part of, and we have been trying to develop some of these recurrent network models that actually perform much better and are more brain aligned than the simple feed-forward models of the time. Okay. Another thing that, so as an experimenter, this is not fully satisfactory. So, okay, I told you that there is some recurrent connection, there is some recurrent computation. Where is this computation, where is this signal coming from in IT, this secret sauce in IT? And if you look at the, circuitry of IT, it's very messy, and there's a lot of ways in which this signal could be arising. So I thought, okay, let's start by ruling out like the stuff that might be slightly easier to address experimentally. So what I thought, okay, there's prefrontal cortex, also previously associated with object categorization type behavior. Let's try and inactivate prefrontal cortex and see what happens to the signal in IT. How important is the prefrontal cortex contribution to IT in solving this task? And does it affect in any meaningful way with respect to our previous results? So what I did, the idea is that, okay, our top-down feedback from VLPFC critical to this behavior. You show an image, the image gets processed through the entire brain, a ventral pathway, goes to the PFC. This is a simple idea, comes back to IT. Let's say this is the default scenario. Now we perturb the system using mucimol injection. Maybe some of the feedback from PFC to IT is reduced. And we ask, okay, what happens to the previous experiments that we just did? So we implant arrays again in IT, and we ask the monkey to do the same behavior, but now under two conditions, the perturbed and the unperturbed condition. Okay, so we have a previous hypothesis that late solved images maybe are more, influenced by this recurrent signal. So we have the hypothesis that maybe they will be more affected by whatever we are doing with the mucimol injections in PFC. Okay, so this is our neural analysis showed exactly the same. So if you look at the neural, prediction, these predictions are much worse. The deficit was much higher for the late solved images. Same with behavior. There were deficits observed in both sets of images, but the late solved images showed much higher deficits. Again, I'm really summarizing a very detailed study in 2 minutes. So if you want to know about the details, please look up the paper, get in touch with me, and I'll be happy to answer more questions about it. The other interesting thing which we observed was that I also showed you this result where models were predicting early responses better than feed-forward models were predicting early responses better than late response, right? So what did we actually do in the PFC inactivation experiment, silencing experiment? What we think we did was we actually turned down the feedbackness of the model, the recurrency of the model. So in some sense, we actually, not the model, sorry, the monkey. So in some way, we actually made the monkey a little bit more feed-forward like. So interestingly, that's exactly what we also observed in terms of the neural prediction. So now, if we ask the model to predict the neural responses of the monkey, what we ended up seeing is that this gap that was initially observed after silencing, this gap actually reduced. And it reduced specifically because the late responses were slightly better predicted by the models compared to the case where there was no inactivation of PFC. So again, these are converging evidence that what we are doing at the level of PFC here, one of the things that we are doing is we are actually specifically perturbing the feedback that is coming from PFC to IT, right? So again, in terms of model building, what we need now is we need to build better models that actually incorporate this PFC module, which wasn't incorporated before in our previous models. And at the same time, I'm doing experiments where we are recording from the prefrontal cortex to get more data to help these efforts in the community and in the computational neuroscience community. Okay, so the last thing that I want to talk about a little bit is slightly more into the experimental realm. So I'm an experimentalist primarily, but I want to, as I have initially shown you, our research program really wants to influence models and help improve them in the long run. So I'm going to tell you why the previous experiment that I did was not the perfect experiment and why it was actually had a lot of confounds in it. So one of the confounds you can appreciate maybe if I show you this diagram. So this is, let's say this is the part of the prefrontal cortex, ventral prefrontal cortex that you want to, perturb. And this is the part of the IT that it projects back to. So when in the previous study, when I wanted to perturb VLPFC, I did not want to perturb, all these connections that were going out of VLPFC to other areas. What I wanted to perturb, when I injected mucimol, it affected all of these connections. But really what I wanted to do was perturb these specific connections, right? So that was not possible in an experiment like mucimol injection. So this is another type of, methods development that I'm very interested in. I did a little bit of that during my postdoc, and we're continuing doing that in my own lab. So how do you do that? And then you can, gain tools from the rodent literature and keep testing it on monkeys and see what works. So the idea has been tested in rodents, and the idea in some sense is pretty simple. So you will inject a virus, a Cre virus that expresses Cre here, that is taken up. It's a retrograde Cre virus, so taken up by the axons, passed on to the cell bodies of these neurons. Okay. And then you inject another virus here that is a Cre dependent. So it will only affect the neurons that have expressed Cre. And if whatever this DREADS virus does, and I'm not going to explain much about DREADS and everything, but you can imagine like this is a kind of, substance that if you put in will do something to the system that will produce the desired effect, but now only in these neurons that have taken up the Cre from this other area. So in our case, these DREADS virus could, could transfect the neurons in that area to develop receptors that end up, producing less activity in the area, in those neurons, which will serve our purpose. But that's not the point. The point is that in this way, you can develop an intersectional strategy. But it requires a lot of work. It requires us to test these viruses independently at both ends. And this is what I have spent a lot of time doing. We have tested many monkeys, and many viruses work for rodents, but don't work for monkeys for multiple reasons. So we did find a couple of ones that work. And also, again, when you want to use Cre-dependent DREADS, you first want to know if there is, forget about Cre dependency, even a DREADS strategy, does it work in monkeys? So this is also ongoing work, and I actually summarized a lot of our work in this talk from 2021. So if you're interested, you can go to this website and you can see this talk, sort of, summarizing what we have done in this realm. If you're interested, you can do that. Okay, so here I think is my last slide. Many times, like specifically folks who are working with these deep neural networks are asked this question. Yeah, you have shown this complicated model and this sort of like idea that PFC's signal comes to IT and this is the recurrence and this improves the behavior. But what is the role of top-down interaction? And this is very dissatisfying to me, because this question is kind of like really not well posed in some sense. And this question is actually a very common question that has been asked throughout the history of neuroscience. And it has been asked in a very different way. It has been asked in a way, what drives neurons in area X? And I have to tell you that this question is very important if you have no clue about what area X is. If you have no clue where you're recording from, is it a visual area? Is it a motor area? Is it auditory area? Okay, you need to know a little bit about the lay of the land. But at some point, this question becomes a dead end. It is not really, it's perhaps not the best way to keep thinking about the area. So what drives the neuron in area X? So let's start answering the question. What drives neuron in area V1? A typical answer to that would be oriented gabors. Like different orientation produces different kinds of patterns. What drives neurons in V2 would be texture. What drives neurons in V4 would be complex curvature. What drives neurons in IT could be like faces and objects. And now we know, and there has been multiple papers showing that these answers, if anything, are incomplete. These don't really completely explain what is going on in this area. And I would argue that what we need are models that predict the responses of these neurons in these areas. And you can test those models in as many ways as you want. You can, implement as many benchmarks, as many ways of hypothesis testing for those models. But at the end of the day, it is going to be a model that looks like the brain that will be an answer. And the difference between that model and another brain is that we will have access to each part of that model. We will have complete understanding, and in my case, that is a real understanding, a mathematical understanding of complete cause-effect relationship of one parameter with the other. We can write down the equation, we can, and this is true for convolutional neural networks as well. It's just that convolutional neural networks don't really explain much of the brain. So anyways, I think instead of focusing on what drives the neuron, we should be focusing on what types of models, what class of models are best predicting the activity or whatever phenomena that you're interested in. Right. So even the idea that what is the role of the top-down interaction of, rather than asking the question, what is the role of top-down interaction, let's ask the question, what kind of models with top-down interaction best explain the data that we are collecting? And what kind of data can we collect to falsify your favorite model? So let's focus on building these full-scale predictive models for a bit and let go of our infatuation with these words and low-dimensional toy models. And by let's, I mean, we will focus on that. And the rest of the decision is up to you, what you want to do. So here is the lab that really influenced my life a lot. This is Jim DiCarlo's lab, and we had a lot of fun. Thank you to all the lab members for motivating me throughout the years. And so I have recently also started my own lab at York University with a lot of folks who are doing non-human primate work. And we are getting this new nice building to do our work together, specifically focusing on non-human primate visual neurophysiology in this new center. And I thank all my lab members for really making my last couple of years a lot of fun working through this problem together, and all my funding agencies to keep the light on in our lab. And thank you for your attention. Happy to take questions.
Thanks so much for the very interesting talk and being on time and everything. It was very insightful. And I already see some questions on the Q&A, and we can go over them. So the first one is asking for a career advice. They would like insights and tips into joining major research institutes like McGovern at MIT.
Okay, so well, I'll tell you, like I worked at McGovern, so I can tell you how I went and worked there. The first thing I did was I was very interested in the field. So I tried to join as many summer schools as possible. And so there are all these summer schools, specifically like Neuromanage, the one that you're taking right now, where I did connect with the community. And that was the first step. And I think you are already in the right direction in that sense, because you have connected to the community. And I think that's important. And also not just to, connect to go to MIT, is just that you want to also interview these folks at MIT. Like your time might also be worth something that maybe you don't want to spend at MIT. So you need to also know the community a little bit and decide whether you want to spend time in this community. So I went to this summer school at Cold Spring Harbor, and I was at a talk, and I really liked the talk, and I looked up the papers that they were referring to, and I found a lot of paper from Jim DiCarlo's lab. And so I emailed Jim, and a lot of this is, serendipitous luck, like what kind of expertise or what kind of trainees they were looking at the time, and everything matched up. And I think, I want to answer this question a little bit also from the point of, like, you're asking specifically about maybe like McGovern, MIT, like you might have something in your mind, like when you say like, there are a lot of good institutes around. So keep your mind slightly open. But I think if you show interest and if you really are motivated to do this work forward, I think keep applying and you will get in. Sometimes, like, I think life's unfair because a lot of people have issues that are not related to their skill set, that end up influencing their career. Like, many students of mine cannot go and present international conferences because they simply don't get visa. And whenever you are lucky for some reason, like you're someone that gets cancer and you don't, that's your luck. I just treat my life that way, because I feel like it will be very depressing to just think about like what kind of factors are coming in and influencing and helping others and not influencing. Some of it you can make some changes, but some of it you cannot. So for you, you're doing the right thing. You're here, you're motivating yourself to study more, learn more about the system, and when the current opportunity comes in, apply, engage. There is no wrong question. Keep asking. If you ask somebody that I want to work with you, I really like your work, give me an opportunity, that's not an annoying email. So I think if you're really interested, if you show interest, you will get heard at some point.
Mhm.
I assumed a lot of stuff about you during this answer, but hope it helped a little bit.
Yeah, thank you so much. These were all very great advice. So the next question is about with all like like biological brain utilizes recurrent architectures to solve problems like, for example, maybe like high energy efficiency, whereas larger scale AI systems, such as those that are using transformers, rely on massive feedforward architectures and largely overlook the potential of recurrency, right? Like we know that in the AI field, they don't necessarily care about recurrency. So the question is, how do you interpret the implications of these contrasts for the future of AI and also neuro AI?
Okay, that's a good question. So my personal opinion about this is that currently I don't think neuroscience has been influential for AI in the beginning, but I think currently it's a very weak link. Like, I don't think somebody is going to read a paper from Journal of Neuroscience or Nature Neuroscience and have an idea, and that would change like the course of AI research in the immediate time. So I think at some point these two objectives will diverge, or the products will diverge, because AI is optimizing for something different than neuroscience. But neuroscientists are not very good at building large scale models. So neuroscientists, I think, unless the training protocol and it's already changing quite a bit, unless that changes, we are sort of dependent on neuro NeurIPS and iClear to come up with some models that we can download into PyTorch and play around with. So I think that way, to me, like inflow from like AI to neuroscience is at the moment looks like way more productive, and I think we should capitalize on that. The place where I think maybe neuroscience or mostly behavioral science might play a big role for AI in the future might be if you want to make solutions that are very specific to humans, and those I think might require some understanding of how humans behave. Uh, I don't think to excel in something, the idea that, oh, humans can play chess or humans can, play Go, so let's understand how humans do it and play Go better. I think by doing that, you're going to only constrain yourself to, be as bad as humans in all of those things. So I think in some sense, if AI is trying to supersede humans, having humans as a final goal or some sort of constraining factor might actually do more harm than good for the rise of AI. But I think that will be interesting, interactions specifically when humans are in the loop to be helped by AI. So then AI might have to specifically, cater to the kind of algorithms that are basically implemented in our brain through evolution. So that's what I feel maybe the one implication of what's happening.
Mm-hmm. Very, very insightful. Thank you. So the next question is about the I think the study that you inhibited PFC, ventral PFC, the attendee, which is anonymous attendee, asks, when you mentioned selectively inhibiting prefrontal cortex activity, was it only inhibiting it experimentally, and therefore not including it in the neural circuitry, in the data input to the model, or is each brain region modeled? If I understand the question correctly, it's about like, did you do modeling with like each region being in the model, and then you inhibit that region, or was it just like experimentally with injecting the proper
Okay, so
material?
I'm not fully sure what the question is, but I would just explain the setup again. So we injected mucimol in the monkey's brain and then recorded monkey's brain and in PFC, but we recorded in IT of the monkey. So in the models, there was no inactivation, nothing. The model was just running like the way it was running before. And in the models, typically we consider mapping the model to areas like V1, V2, V4, and IT, not necessarily PFC. So there was no component of the model that was inhibited or inactivated. An interesting, extension of this question or this sort of reasoning could be that ideally you might want to have a model that have a PFC and you want to do the exact same question. Sorry, you want to do the exact same experiment in the model and then ask whether your experimental data actually align with what you see in the model. So, just to test the model, essentially, to falsify the model. So that's another interesting future direction. So for that, you need models with PFC, and you need to know how much of the brain are you actually perturbing, and you need to make an equivalent perturbation in the model.
Just, I have also a question about that experiment. So like, to control for not inhibiting all the connections coming out from the PFC, you did the other approach that you first injected some virus to IT that then traced back to PFC, and then you especially targeted those areas. Would that injection to IT impact the like neural activity in IT?
Ideally, it should not.
Okay, so that's
but when we are recording like when we are doing this experiment, they will also be present during the I would say they would also be present when we are not inactivating PFC. Like we are not inactivating the other, we're not injecting the other drug in the system, right? Like we are not, I would say, when we are not inactivating the DRESS through systemic injections. Maybe one way of answering your question could be that you could do the same experiment with just injection of the viruses, but never actually activating those viruses. So you keep them in the system, but you never load them up in like with the activated drug that actually makes. So to see what the virus just does in the system when it's not being activated. If that's your question, I'm not exactly.
Yes, like
because,
but I thought it's a very neat method to like selectively choose those connections, and I'm impressed. I thought like, would that like have an effect on like, because before you would inject to PFC and see its effect on IT, right? IT was intact, but then the other method, you are also injecting a drug to IT.
No, you're not injecting the drug. You are injecting two viruses in two areas, and that's only one surgery, and then it's done. You never perturb the system anymore. So now, whenever you're recording your control condition, you're still, have those viruses in IT. They're just not activated because that's not to activate this whole, intersectional strategy. You have to inject another drug in the bloodstream of the monkey.
Oh, okay.
So that takes you to the perturbed condition where only those projections are affected. And in a non-perturbed condition, the viruses are still there. Like the viruses manifest as like receptors in the neurons, which are otherwise inactive. If you don't put the activating agent, they don't really interact with the activity of the brain.
Okay, yes, that makes sense. Thanks. Yep. Okay, so the next question is from Jay Lee, and they ask, Is there some properties of visual stimuli that test the impaired top-down modulation of PFC? Like monkeys should recognize the context and understand the object. How did you synthesize the images?
That's a very good question, actually. So I think that that is sort of an experiment, a project that you can do, which is so we chose these images, challenge control, just by screening through many images and doing the behavior in monkeys and humans and models, and just figuring out which images are, you know, more like better solved by monkeys and humans compared to models. So that's how we got to those images. You know, there is a recent strategy from Krieger-Skorda's lab and Tal Golan's work about controversial stimuli, which is like you have two models and you or multiple models and you find stimuli that disagree between these models. So what I showed you for challenge and control is kind of a controversial stimuli between monkeys and models. Like these are images that are very differently behaving, or the models, the two species act very differently for these kind of between monkeys and models. You could synthesize images now that produce the same type of behavior for a recurrent system and a non-recurrent system. That might be a very interesting project. So you develop a model that is a recurrent model, you develop a model that is a feed-forward model only, and you try and synthesize images that are specifically solved better by having a recurrent model compared to a non-recurrent model. And that way you might start asking these sort of questions about what factor in those images were driving. I mean, you asked this question about context. I think that's a very broad term. So maybe like, you know, whatever I show you might somebody might say, well, that was context. So I think stay away from these, like, you know, one word terminologies. Like these are all images, and let's think about like strategies to figure out these images that are like richer in dimension than maybe like one or two specific. I mean, it could turn out to be that contextual, you know, manipulation induces these kind of effects. In fact, recently we are, you know, pursuing a sort of like direction like that, but that was not specifically to synthesize images of this sort, but to understand more about contextual reasoning. But I like the question. I think you should try and see if you can synthesize images that are specifically benefiting by a recurrent network compared to a feed-forward network.
Mm-hmm. Okay, thanks.
Yeah.
The next question is by Arjun Puri, and they want to know, or they want more explanation about the role of the decoding model accuracy and the delayed onset of the spike for challenge images, and like how you made the interpretation that like that delayed in decoding for challenge images are related to recurrent processing.
Okay, so let's take this here. So I don't think there was a delay in the onset of the spike for the challenge images. That was actually one of the points that I made. So let's go with the decoder. So what is, I don't think it will be possible to explain here how the decoder works, but you can, so the decoder essentially is a, you can think of this as like can you see my screen here with like, I'm trying to draw here. So let's say these are some neurons, and I will join these neurons with some weights to some node, and let's call that, you know, a face node, and let's join another node, and let's call that like a dog node. Now this is an image of a face. So when I show this image of a face and these neurons behave in certain way, respond in certain way, these lines are basically some weighting function that you have derived through some fancy algorithm. You come up with the best possible weights to attach it. If your model is correct, the weights will work in such a way that this node will have a higher activity than the dog node. And that's the very basic idea of like what a decoding algorithm should be doing if you have trained it correctly. So what I'm showing is that around this timeline, like until here, there's no activity. So the decoders are zero, which almost means that if you can think of this as like face versus dog task, if this output is 0.5, this output is also 0.5, which means that they really are not that different, which can make sense that there's no difference in these two. Whereas when you start having some activity in the neuron, there's some pattern here. There's some pattern of activity happening here. now all of a sudden you're very accurate, which means now you can actually differentiate between a face and a dog, right? So this is what happens for a control image. Okay, so that's exactly the point for a control image. Now imagine you have, I'll show you the one for the challenge image. Move on, the challenge image. Let's see. Oh, did I not? Okay, so now let's look at the challenge image. So the challenge image at the same time here, sorry, I have to bring this up and then put my, so the challenge image around the same time here is quite low, but there is activity around this time. So this is what I was saying, that there is no lack of firing in this time, right? So there is still spikes, but there is this decoder that you trained up was not working. Somehow, all of a sudden, for certain different kind of pattern, I'm drawing really poorly, the decoding accuracy went up. So the question is, what happened between these two patterns? Like, what made this pattern of firing change in a way that the decoder that was not working before all of a sudden started to work? So, and I think the hypothesis was that maybe that's what feedback is enabling, that it is changing the pattern of the response in a way that your linear classifiers will start working. Again, one main thing you should remember is that all of this that I'm saying are not facts about the brain. They're hypotheses about how the brain might be working. And all of them needs to be tested, and you should keep testing these hypotheses. They're not like, hey, this is how the brain works. Any of these fancy algorithms, what they're doing, they're called readout models. They're proposing how a downstream region might read out the activity of IT and produce behavior. So that's what the purpose of these decoding models are. So just because decoding models are, producing high accuracy doesn't mean they're working in the same way as the brain. So there are other papers that we have worked on where we show how you want to test these equivalences of, like, how similar are model outputs to behavioral decode outputs from the neuron.
Okay, thank you very much. Okay, so I think you talked about like the viruses, like we discussed that. So I skipped that question and I go to the next question, which is by Mahdia, and they ask, why do you think there is a hard wiring between IT and PFC? Basically, why you decided to choose to inhibit PFC, and you think that there is a like direct connection from PFC to IT, and but like, for example, why there isn't there an intermediate region connecting those together for the experiment that you did?
So one thing is that, so I think you're talking about the virus injection experiments. So the only thing that we have tested is whether the technique works. We haven't actually done any scientific study using that technique. We have done the study using mucimol, which just takes out PFC as a whole. Of course, there might be intermediate regions, but there are also direct connections between IT and ventral PFC that I was showing. So the method, at least the way I presented, assumes that there are these monosynaptic connections between these two regions. So of course there are intermediate regions that also join these two regions, but there are previous studies that have shown, tracing studies that have shown that there exist monosynaptic connections between these two regions that actually enables our method, or enables the success of our method potentially. The second question was that, can you use the same technique to do optogenetics? Absolutely, yes, that's true. The only reason to do chemogenetics compared to optogenetics is that, at least for us, would be for optogenetics, you have to deliver light to inactivate or activate the neurons that you have transfected. And sometimes you cannot, push light in deep parts of the brain. So for that purpose, chemogenetics is actually quite good, because you can inject anywhere, because the way you activate those receptors is through systemic injections, which means you inject in the bloodstream of the animal. What you lose out in chemogenetics is actually you lose out temporal specificity. So for optogenetics, you can switch on and off the light very quickly. In chemogenetics, you have very slow acting, like hours, 2 hours sort of time course of like something gets activated, then it takes a while to get deactivated. So it's more like Musimol in that sense. But there are so many questions at the larger level that are still unanswered. So I don't think that there are even any models that make temporal level predictions. So in some way, I don't see that much of a value of the temporal aspect of optogenetics over chemogenetics. But of course, these are two different types of information that you are perturbing. So there might be very interesting results from optogenetics that are not really retrievable from chemogenetic experiments like dreads.
Okay, thank you very much. So the next question is about, can you discuss tips for fostering successful collaborations between experimentalists and computational neuroscientists?
Yeah, my first thing is like stop asking for data. It's like something that experimentalists hate because it almost, and this is because science at the end of the day, no matter how altruistic we pretend to be, is a social activity. It's a social thing. And I think a lot of times, at least in my own career, I've seen is that experimentalists are treated as like this sort of data providers, like let me do all the thinking and you just get some data and give it to me. And many times in that what has happened is that experimenters would do, because all experimenters at the end of the day, they actually want to make an inference. So they're also interested in knowing the answer to the question that they have asked, right? So what ends up happening is that they will answer their question, which was probably the most important question for that data, because that's how the experiment was designed. And then they give out some scraps to the modelers to play with. So many times the modeling papers that come out are like secondary, third level analysis on some primary experimental data that was designed, and oftentimes that's why they're suboptimal. So I think the way to foster successful collaboration between experimental neuroscientists and computational neuroscientists, by the way, I don't think these terminologies will stay so separate. They're sort of like blurring already, but you still need experts in different fields. But what I think is that you need to come in early. You need to start at the level of the experimental design. And I think for that, there has to be labs that foster that kind of environment. So I was fortunate that I was in Jim's lab, where I met Puya Bashivan, was also a computational person. And then we did this project together where there was a perfect sort of balance between experiments and modeling. And we both did things that just by ourselves we could not do alone. And I think those are the places where collaboration actually makes sense. Otherwise, yeah, after but you put the data somewhere for others to play around with. That's one way of collaboration. That I don't think that was the question. Also, one other thing that I want to point out, that initially, yes, there was this idea that you have to put your data out and other people should work on that data, and I think that is still very valuable. But more and more, I'm thinking about benchmarking models and understanding which is the best model of the visual system, primate visual system. It actually becomes very important how much data you give out publicly and how much you hold back for testing the models, because we have enough tools now to just train our models on the existing data and predict all the data that we have. So there are interesting considerations that needs to be made as well. But again, these considerations are better made by computational folks, honestly, than experimental. So again, there's a lot of room for collaboration, but stop treating experimentalists as sort of data providers and engage with them as scientists. And I think you will get more data and more collaborations out of it, is my honest opinion.
Okay, thanks. Okay, so the next question is about what drives neurons in area X if a stimulus features are not the correct way to look at it, and would focusing on fundamental mechanisms of neural bioenergetics be a better way to look at it, or would that be too general to be useful?
So I think the question is that, to me, the important part is that how are you improving the model? What model are you actually building that is explaining the data that you're collecting? So if knowing what type of images make firing rate go very high in a model, or sorry, in a neuron in an area, if that information helps you, then it's actually very good information in some way. In the same way, if some assumption about bioenergetics, like you need to have lower wiring cost or something, if that helps you build better models, then that's a good approach. But at the end of the day, the test is in how good the models are predicting the neurons, not how good your, how smart the hypothesis sounds like or how cool the images look like, which is often the case. So I think at the end of the day, you can have a very cool hypothesis about bioenergetics or a very cool system of like generating images, but if the models don't predict the neurons or the behavior, it's useless to me. So I think you should anchor it on the model and then do all this other. And by the way, there are multiple papers coming out from Dan Yamin's lab, Kalanitz, Gil Spector's lab, many labs, Jim DiCarlo's lab, where they are using bioenergetics principles, similar principles, to actually build models. So these are relevant research directions for the field. By the way, I just want to also make sure that we stand like on the shoulder of giants. Like all this previous work where people have shown that how different features of the images impact different neuronal responses. I'm not really trying to say that they were not useful, but I think at some point we need to do something different if we hit a wall. So that's what I'm mostly trying to channel.
Okay, thank you. And the next question is about how would you place Vienna model, which was developed to model how brain processes art?
How would I place them? I'll place them as a hypothesis in the bucket of multiple hypotheses that exist in terms of how the brain works. Again, I don't consider like that there are multiple questions about like how does brain process art, like how well is art, responses to art, how different is this across humans? Are we talking about like one individual humans or experts art? Like maybe the paper that I've not read the Vienna model paper, but I'm assuming that there are a lot of interesting questions just from a simple behavioral point of view to answer in that case before even going into models. But it could be an interesting model, and I know this is like sounds kind of interesting and hot, right? Like so, yeah, I would place that as one of the hypotheses of how the brain might work.
Okay, so thanks. So the last question is again, I think, a career advice one. They say that I'm a PhD student, a TA in Neuro AI course. What should my profile have if I want to join the Carlo Lab or co-lab as a postdoc?
Well, the Carlo Lab or CoLab should have funding for you, or you should have your funding. So that's, I think, practically a very important point, because without that, we could all have a lot of, ideas, but they will fail. So that's the key, one of the key things here. The other thing I would say is that, the last 2 years or 5 years of work in these labs kind of gives you a sense of where this lab is headed. These labs are headed. And so I think if you study those and if you have interesting ideas, that might help. But at the same time, one thing I want to channel a little bit as a professional development thing is that, like, and I understand the point, like I was also in the same boat, and I also thought about these issues in the same way that maybe you are doing, which is that you are thinking, how would they take me? I want to also motivate you to think like, why should I work with them? Especially in computational neuroscience postdoc, like you will get triple the salary if you join an industry. So when you decide to not do that and come to MIT or York or any other place to work, you're making a big life decision. So you need to evaluate us and know and understand, like, why should you spend your precious time doing the thing that. So I think that's important for you to actually figure out that the kind of stuff that we have been producing over the last 3-4 years, are those exciting to you? And I think if those are exciting to you, that would channel itself in any communication that you have with us. So I think that's really the key. You have to be in these kind of cases, like a career where the rewards are not that regular, very, very sparse, but there are definitely a lot of rewarding moments. So So I think in these kind of cases, you have to be really truly motivated. And I think, again, for your profile, of course, like, you might have all the right kind of interest and not have the skill set to do something. So for specific, more practically, I think, currently, at least in the lab that you have mentioned, my lab or Jim's, I think we would say that having a very, solid computational, base is important, like to know how to code, to at least know either Python or MATLAB or something that basically could help you develop computational insight is probably critical, unless you're proposing to do a very interesting experimental project, which is also probably true in both labs. So it's open, just email and explore. So yeah.
Okay.
But treat yourself more important, like give a little bit more importance to yourself.
Yes, I agree. Yes, you're all qualified, and you should get in touch, explore the opportunities, and find the thing that really best fits your interests. Um, Thank you, Ko, for the wonderful talk. I really enjoyed the discussion. We had
Thanks.
many questions, very interesting questions, and a long discussion. It's like 10:37 already.
Cool.
And it's late night in North America. So thank you very much for joining us today, and very insightful talk, discussions at the end, career advice, everything. I'm sure like the students enjoyed it.
Yeah, I had a lot of fun. Thanks for inviting me.
Of course.
Hope you had a very good summer school experience. And if you see me in a conference or anywhere, like, say hi. And yeah, if you want to know what the lab's doing, you can follow our lab website, or usually I would post on X or Twitter, previously Twitter, so you can follow us there. But like, yeah, just email, be in touch.
Okay. Okay, great. Thanks everyone for joining. Have a great
Bye-bye.
night or morning. Bye.
Take care. Bye-bye.